everyone. Ayan. So, good evening.
Good evening,
Okay.
ma'am.
So, okay. So, we'll have our CEE review for Pharm Analysis 2. So, this is your quality control or PharmChem 204. Sige, let me share my screen. Yan. So, visible na si shared screen.
Yes po, ma'am.
Okay, thank you. Sige, so we shall start, no? Our
Recording in progress.
So first, we go to the key terms or key terminologies used in quality control or quality assurance. So when we say quality, no, so the totality of the features of the product and its conformation to specifications, no? So 'di ba when we see a product, sabi natin maganda ang quality based on the overall features, no? The color, the odor, the taste of the drug or the effect of the drug, no? The side effects, the safety, the effectiveness, no? The totality of the whole product and its conformance to specifications, no? So according to FDA, according to USP, ano ba yung standard natin for this product? So does it meet the standards? So if yes, then it has a good quality or it has quality. Okay, so in achieving quality, we need your quality assurance. So keywords, sige, keywords lang tayo class, no? So sa quality totality features, ayan, quality assurance, our keyword is the sum total, no, of organized arrangements. Meaning, lahat-lahat ng efforts ng company that is intended to keep the quality of the product, no? From the design of the building, from the appointment of employees, no? Lahat ng decisions ng company to produce consistent quality. No? So that's quality assurance. No? The sum total of the organized arrangements with the objective of ensuring quality. Okay? And guiding quality assurance is our CGMP. So what does CGMP mean? Sige nga, what does CGMP mean?
Current manufacturing practice po, ma'am.
So your current good manufacturing practices. No? So that guides your no, in the decisions that they make. So CGMP is based naman on PICS. So PICS means, ayan, nakalagay na dyan, your pharmaceutical inspection, convention, or cooperation scheme. Ayan. And then, so you have CGMP, nasa loob niya si QA, no? And then sa loob ni QA, we have your QC. No? So si QC naman, mas specific ang role on the testing, sampling, and documentation of the product testing. No? So we have your quality control. Okay, so ayan, you have your key terms for different areas of QC. QA versus QC, no? So when we say QA, 'di ba, mas malawak, no? Mas malawak ang kanyang coverage. So assures that policies are followed, no? And cooperates with your regulatory agencies, gumagawa ng SOP, and we have audit and monitoring. So ang audit dito class, this is yearly, ginagawa yan siya sa isang company to check if fina-follow ba ng mga employees yung protocols that are set by the manual. No? So, fina-follow ba? So, that's your audit. O, ito, gina-follow ba ni na SOP? Meron bang mga deviations? Mga ganun. So, you have your audit. So, hindi yan audit na yung sa pera. So, audit yan to check the obedience or the compliance to policies. Okay? And then, so QA is focused on the process. No? So QA, ang focus niya ay ang process. So the whole process of producing the product. Whereas si QC, ang kanyang focus ay ang product mismo. No? So testing the product, quality control of the product, the environmental conditions wherein the product are made. No? So si product ang focus ni QC, whereas the process of producing the product ang kay QA. No? So mas malawak yung kay QA. Okay, so different functions of QA, and these are the functions of QC. Mostly on the product, ito mostly on the process that the company does for the product. Okay, so next we have your method validation. So in QC class, we do different procedures, and the procedure should be valid. What do we mean by valid? No, pag sinabi nating valid, they perform their intended purpose. So let's say, for identification, nakaka-identify ba talaga ang procedure na to sa presence of, let's say, paracetamol? Quantitative impurities testing, does it quantify accurately, precisely the impurities present? No? So, we can make sure that the procedure is valid through validation. No? So this is your method validation. So ito, summarized in this table are the different parameters na kailangan mong i-test per type of procedure. So let's say identification procedure. So you just have to determine the specificity. Specific ba siya sa inyong target analyte? Then for assay, lahat except limit of detection and limit of quantification. And then for impurities, na depende sa impurities. Kung limit testing lang, when we say limit testing, you just check if madami or konti. No, below or above the limit. So yun lang yung tanong. Hindi mo kailangan i-quantify. Hindi mo kailangan malaman kung ano yung exact amount. Gusto mo lang malaman if pasa ba siya sa limit or hindi. So, ano lang, konti lang yung kailangan i-validate, no? The specificity and the limit of detection. Whereas sa quantification or quantitative impurities testing, gusto mong mag-quantify, kailangan accurate, kailangan precise, kailangan linear, and etc. No? So you have yes, 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 lahat except LOT. Okay, so ayan, you have your method validation parameter. So you already have a copy of this, no? So brush up lang natin yung different concepts. Okay, so in QC, in the manufacturing company, we have three main areas. We have your RMQC. So ano ang meaning ng RMQC? Anyone? RMQC, so quality control for ano yon?
Raw material quality control po.
Your raw material quality control. And then we have IPQC. IPQC. How about IPQC?
Process quality control.
You have in-process quality control. Very good. And then lastly, we have your FPQC. FPQ.
Finished product po.
Okay, finished
Quality
product quality control. And may additional dyan, pero hindi masyadong major. You have your packaging material. Yeah, packaging material quality control. So parang additional. Okay, so unahin natin si RMQC. Okay, so for our RMQC, no, so in order for us to have good product, we have to have good raw materials, no, good starting materials, no. So hindi ka makaka-expect ng magandang product kung substandard yung materials or ingredients na ginamit mo. So first, we have to perform identification tests, no. So identification tests could be chemical, physical, and biological. So first, chemical tayo. So chemical involves visible reaction. So precipitation, color change, evolution of gas, or any specific identification reaction. No? So based on a chemical reaction, ang ating chemical type of identification method. Okay. So, whenever there are impurities present in our raw materials, we have the following category. So when we say adulteration, no? So debasement of the article. May dinagdag, may tinanggal, may nago. No? That's adulteration. So adulteration could be present in different forms. No? So we have sophistication, inferior material ang dinagdag. So let's say dapat type 1 glass, ginawang type 2. No? So addition of inferior material. You have your sophistication. So dapat ganitong brand, ginawang generic or ibang brand na mas mura. So that's your true adulteration or sophistication. Okay, next we have substitution. So sa substitution class, iba na talaga, no? So keyword is entirely different than the one requested. So substitute ka talaga, no? So instead na methyl cellulose, ginawa mong carboxymethyl cellulose. So entirely different from the one requested. And then we have admixture. Admixture class is, ang keyword is accident. No, hindi mo intended na gawin, pero dahil sa katangahan, sa aksidente at carelessness, no, nadagdag siya. No? So parang kay Professor Powerpuff Girls, no? So hindi niya intended na malagyan ng other substances para gumawa ng Powerpuff Girls. So that's admixture. And then we have spoilage. So keyword kay spoilage, microorganism. So microbiological degradation, no? Pag degrade dahil sa presence of bacteria, fungi, virus. No? And then lastly, we have deterioration. So sa deterioration, keyword, due to the environment, presence of insects, or any other environmental factors, no? Dahil sa heat, dahil sa sa humidity, sa temperature, no? So sa moisture present sa environment, no? So kaya siya nag-deteriorate. So maganda sana, pero nag-deteriorate dahil sa environment. So in all of this, no, adulteration happens, no? Meaning nagbago ang original product mo. Okay? So, for different reasons. No? Depende sa reason ang kanyang type of adulteration. Okay? So, ayan. Okay, next we have physical method. So, physical method, no, nasa pangalan na, we involve physical constants, no? So, specific gravity, the density, the refractive index, and the optical rotation. No, bakit? Bakit pwedeng gamitin si physical method? Kasi, dapat itong mga constant values na ito, constant nga. So, if pure ang substance or it is the correct identity, no? Hindi magbabago yung physical constant value. No? So the specific gravity of water, for example, is 1. No? So hindi yan magbabago ever kung pure si water. Pero kung masagulan si water of other solvents, then the specific gravity changes. So somehow a change in the values of these physical constants, no, will tell you that there is something wrong, no? May impurities na nangyari or involved. So isa-isahin natin yan. So first, specific gravity, no? So defined as the ratio of the density of a substance to that of a reference substance, no, at a specified volume and temperature. So at what temperature natin kinukuha si specific gravity? Sige, at what temperature? Sige, any guess? At what temperature natin minimeasure si specific gravity? Using your pycnometer. Sige, may idea? Your room temperature. So what is the room temperature? At 25 degrees Celsius, no? At 25 degrees Celsius. Except, I think, alcohol. Si alcohol, I think, at 20 tayo. Okay, so that's your specific gravity. So, what instrument? So, importante, you know the instrument class na ginagamit to measure. So, specific gravity is measured using your pycnometer. Ito siya. And or your Westphal balance. No? This is your Westphal balance. Isulat ko. Okay. Yan, your Westphal balance. So both can be used to measure the specific gravity of different liquids. Okay, next, we have your refractive index. No, so refractive index describes how light propagates through a medium. No, so mag-bend ba ang light? Mag-refract ba ang light? No, so that is specific for a substance. Okay, so we use your Abe refractometer. So this is your Abe refractometer. Again, kung pure ang substance, hindi yan magbabago kung ano yung refractive index niya. But kung impure, nag-deteriorate na yung oil, for example, magbabago yung refractive index. Next, optical activity. So, that's your ability to rotate plane polarized light. No? So, ito yung dextro and levo. No? Yung dextro and levo. So, if it rotates it to the right, so positive or dextro, and then if it rotates it to the left, no, ang light is rotated to counterclockwise or to the left, negative or levo. No, so take note, yan yung small letter D at small letter L, hindi yung malaki. Kasi yung malaki, it's related to the position of the OH. So yan yung maliit na D and maliit na L. No, so that's your optical activity. Dextro, si D, positive. And si levo, negative. No, levo rotatory. Meaning it rotates to the left or counterclockwise. So, it is using your instrument polarimeter. No? So, this is your polarimeter. So, dito class, ilalagay mo yung sample mo. Tapos, dito mo titingnan. So, dito mo titingnan kung yung light is naro-rotate to the right or to the left. Okay. Yan. So, meron tayong polarimeter sa school. Pero I don't think functioning. So, nakagamit kami ng polarimeter sa Manov na. Okay, next, we have your melting point determination and solubility. So, sa melting point class, that will tell you the identity also or the purity. No? So, pag nagbago yung melting point of a substance, example, kay aspirin, it will tell you that aspirin may be degraded or nasira na si aspirin or hindi yun aspirin. Hindi siya pure na aspirin. No? So the melting point is also a good identification check. Okay? So memorize, no? So hindi lang sa class na ito or sa PharmCal, no, sa module 3. No, sa practice of pharmacy, present din ito. So, memorize, ayan, conversion of Celsius. So, si Nancy, nawalan ng 160. Okay, so, si Nancy, may 5 pesos, nawalan ng 160. So, kung sino yung wala, i-substitute nyo lang. Or, sino yung meron, yun yung i-substitute nyo rather. So, 9C is equal to 5F minus 160. No, so you can both find the Fahrenheit and Celsius, whichever is missing. No, so yan lang for the conversion. No, very basic but always yan important. Okay, so ayan for physical method. Last, we have for instrumental, we have your different instruments, no? So that's the modern methods of analysis, no? Instrumental. So we have your UV-Vis, no? Your IR, NMR, and HPLC. Although NMR class, it is seldom used kasi it's impractical, no? Mahal ang instrument, tapos konti lang ang NMR experts sa Philippines or even in the world, no? So there are only a number of people that are expert in reading NMR results. So, hindi siya masyadong ginagamit. Ginagamit lang siya for research purposes. No, extreme research na. Okay. So, yan. You have your instrumental method. So, isa-isahin natin. So, we have your IR. No, IR is the first identification or the tawag nito, gold standard, no, for identification method. No? So, meaning siya yung gold standard. So, if sinabi niya na yan ang identity ng isang sample, 100%, that's the correct answer. No? Siya yung gold standard for identification. Okay. So, in IR spectroscopy, we check the fingerprint region. No? So, what is the fingerprint region? So, this part. No? This is your fingerprint region. So, titingnan natin if this part matches this part. No? So, ima-match yan. Now, if it matches 99 to 100%, then they are the same sample or the same compound. No? So, sa IR class, each compound has a unique fingerprint region. Meaning, para siyang thumb mark. So, parang fingerprint. No? Parang thumb mark ng isang tao. No? So, that is unique for you. No? So, parang ganun lang din sa IR. Except enantiomers. No? Enantiomers, pag enantiomers class, pwede silang pareho sila ng result sa IR. But kung hindi enantiomers, generally, kung magkaibang compound, magkaibang fingerprint region. So, it's very reliable in identification testing. Okay? Ayan. Next, we have your UV-Vis. Si UV-Vis class, hindi masyadong trustworthy. So, less trustworthy. Bakit? Because its result, no? So, it compares the absorption maxima and absorption minima as well as absorption intensities. No? However, those aforementioned could be the same for different compounds. Meaning, for example, si aspirin, tsaka si paracetamol, there is a chance that they can have the same results in terms of the following parameters. No? Pwede silang magkapareho in that sense. So, hindi siya masyadong reliable. Okay? Unlike kay IR, kung magkaibang compounds talaga, magkaibang fingerprint region. No? Kaya it is less trustworthy. Next, we have TLC. So, same goes for UV-Vis because it only compares the RF value, the color of the spot. No? So, kung same ng RF value, baka pareho sila ng identity. But you are not sure. No? Kasi meron pa rin compounds that can have the same RF value but different compounds. No? Magkaiba sila, pero pareho lang sila ng RF value. May mga ganong instances. Kaya hindi pa rin siya definitive. No? Meaning, hindi ka talaga sure kung siya lang yung identification test. Dapat maback-upan pa siya ng iba. Okay? But it's very convenient, very cheap lang ang instrumentation. So, ginagawa pa rin siya. Pero dapat, hindi siya alone. No, sasabayan siya ng iba para pang-confirm. Siya ang pang-confirm. Okay, then we have HPLC. So, sa HPLC class, medyo sumisikat na siya ngayon as an alternative for IR. So, maganda na siya for maganda siyang alternative for IR. So, bakit? Kasi, somehow it is accurate, no, sa kanyang identification parameter. So we have your retention time to be compared in HPLC. No, so kung pareho sila time lumabas na, will tell you they ba siya at 30 minutes minute. Tapos si spironolactone lumabas siya. Then si sample X ay spironolactone. No? So if they have the same retention time, meaning the time na lumabas yung peak, then they have the same identity. Okay, so next is the assay. No? So when we say assay, we check for its concentration or its effectiveness. No? So we have assay. So we can do instrument analysis. So we can use your HPLC ulit, your UV. No? Ginagamit siya for assay. And then we have titration. No? So alam niyo naman yung titration sa Pharm Analysis 1. And then biologic assay. Ito class, when we say biologic assay, we make use of animals or microorganisms. No? So walang pinagbago to, no? Until you take the bucket talaga yun. So you have your animals used in testing of drugs. So we have digitalis for pigeon, no? So sa pigeon class, dako silang heart. So very much observe ang effect ni digitalis on the heart, no? So for your pigeon. Insulin, we test it on rabbits, cat, no? We have your glucagon. No, you have your glucagon. Okay? Oxytocin, so ang ating mnemonics dito is manoxytocin, no? Yung manok, oxytocin. Okay? And then rat, vasopressin, corticotropin, so also rat, pwedeng male, pwedeng female, tubicurarin sa rabbit, also metocurine sa rabbit. Ayan. Then we have chorionic gonadotropin sa female rat. So, bakit female? Ano ba tong chorionic gonadotropin? Ito yung tumataas pag buntis ang patient, no? So, ito yung nade-detect sa pregnancy test. Pag mataas ang HCG mo, muna ito mag-double line. No? So, of course, ang gagamitin mong animal is babae, no? Female rat. Then, we have sheparin, no? So, sheep for heparin. So, for heparin poisoning class, we treat it with protamine. So, same lang, sheep pa rin. Actually, it's the sheep's blood, no? Because heparin is an anticoagulant. And then we have your cod liver oil, no? Tested on rachitic rat. No? So, what do we mean by rachitic? No? What do we mean by rachitic? So, rachitic rats, these are rats that are may rickets, no? Usually, they are deprived of sunlight. Sige. So, anong vitamins nga ang involved or connected kay sunlight? Anong vitamin? Anong vitamin? Vitamin D. Very good. No, vitamin D. So, ang mga rachitic rats class, kulang sila ng vitamin D kasi hindi man siya gipainitan, no? So, the cod liver oil is rich in what vitamins? Vitamin, the oil-soluble vitamin, vitamin A, black dapat. Vitamin A, and vitamin B. No? So kung na-ayo, no? Kung na-cure ang rachitic rat, meaning effective or madami si vitamin D sa cod liver oil. No? So cod liver oil treatment or tested for rachitic rats. No? So if the rickets or the condition of the rats became better with cod liver oil, then somehow you can assume na dahil sa vitamin like cod liver oil. Okay. Next, we have other vitamins and their assay. Quantitative assay ito. So, quantitative, instrumental. No? So, meaning using instruments such as UV-Vis, HPLC, GC. Ayan. So, for vitamin A, we have your spectro. Vitamin D, HPLC. Ayan. So, memorization na lang ito, class. No? So, again, Hindi ito nagbabago. Kahit board exam, ito pa rin yun. So, might as well. Okay? So, vitamin A, sulat lang natin yung mga other names. Vitamin A is also known as vitamin A, other name ni vitamin A, retinol. Very good. No, ito yung mga nasa. Okay, retinol. So, ito yung mga nasa anti-acne, no? Retinol. Sa skincare. Vitamin D. How about vitamin D? Anong other name ni vitamin D? We have your calciferol, no? Or calcitriol. So the active form of vitamin D is calcitriol. Pero yung product sa market is calciferol. So sa body natin, kino-convert natin siya to calcitriol. Then vitamin E, of course, nasa Myra E, no? Your vitamin E. See?
Tocopherol.
Very good. Your D-alpha tocopherol. Okay. And then vitamin K. Sino si vitamin K? Tested using spectro. Si menadione or phytomenadione. Okay, then we have niacin. So this is vitamin niacin. Niacin, vitamin B. Anong vitamin B? B vitamin siya.
Vitamin B3.
Vitamin B3. Very good. So B3, si folic acid. B.
B9 po, ma'am.
Very good. B9. Pantothenic acid. Pantothenic B.
5.
5. Very good. And of course, cyanocobalamin B12. B12. Okay. So, ayan. So, these are your vitamins and how they are tested. So, pwedeng gumamit ng microorganisms such as your Lactobacillus plantarum and Lactobacillus leishmani. No, Lactobacillus yan yung L. Okay. Sige. Other vitamins, we have the following. So, B1, 2, 3, 2, 6, 12. Yan. And other drugs and how they are tested. So, memorize na lang. Okay. So, next, we have microbial assay. So, sa microbial assay, this is usually tested for your antibiotics. No? So, sa antibiotics. So, microbial assay for antibiotics potency, we can do cylinder plate or we can do turbidimetric. No? So, sa micro ninyo, yung Kirby-Bauer, that's your cylinder plate, no? To measure the zone of inhibition. And turbidimetric, yun yung MIC. No? So, using your UV-Vis spectrophotometer. Okay, so for preservatives, ito yung chine-check natin na mga bacteria or fungi against preservatives. So preservatives, dapat effective siya against Candida, against Aspergillus, against E. coli, Pseudomonas, and Staph. So meaning na prevent niya yung growth ng mga organisms na ito. Okay, so that's for your preservatives. For your antibiotics, no? So if ito yung antibiotics nyo, ito yung mga organisms na dapat effective sila. Okay, so for amphotericin B, nystatin, it should be inhibitory for Saccharomyces cerevisiae. No, pag cylinder plate. Pero pag turbidimetric, iba na din. No, so si Saccharomyces cerevisiae sa turbidimetric, for candicidin na siya. No? So, ito siya for your reference lang. I won't be getting any from this. Pero this is for your reference kasi most of the time, this comes out sa board exam. So, for your guidance lang. No? Copy nyo na lang yan. Okay? Sige. Next. So, we have identification. Next. Okay? And third, we have your limit tests. So, test is for your impurities. Okay. So, we have different types of impurities. We have chemical and biological. So, pag sinabi nating gross, di ba? Pag gross, malaki. No? So, visible, nakikita natin impurities, no? So, for example, no? Yung mga buhok, yung mga may dumi, no? May alikabok sa pagkain, no? May langaw. Yan. So, that is your gross impurities. And then have chemical impurities, so yung mga ions, no? Mga sodium salts, heavy metals, degradation products, no? Those are chemical impurities. And biological impurities, of course, mga microorganism. No? Gens, cellular debris, so bacteria na mga products or metabolites, those are biological impurities. Okay, so for chemical impurities, these are some of the limit tests. No? So for arsenic, we can use diethyl thiocarbamate, we can use spectro. Then we have calcium sodium chloride, use flame photometry, so on and so forth. So usually class, some of the tests dito are precipitation, based on precipitation. So example, kay chloride, no? Your chlorides precipitate silver nitrate to become silver chloride, no? Which is a precipitate. So, kung may precipitate na na-produce, meaning may chloride ka. Kasi na-precipitate mo si silver into silver chloride. Same goes for sulfates. No? For sulfates, we test it with barium chloride. So, si barium chloride will react with sulfate to produce barium sulfate. So, barium sulfate is also a black precipitate. No? So, kung may nag-precipitate, meaning may sulfate present. Madaming precipitate, madaming sulfate. No? So somehow the precipitation confirms the presence or the amount of impurities present. Okay, so ayan, you have your different impurities, no? Yung mga naka-blue and then sa white, these are the reagents that could detect these impurities. Okay. Next, impurity involve water, no? So again, di ba water is water could attract microorganisms or could trigger instability, no? Pwede niyang ma-change ang stability ng product. So we have to control the moisture or the water content. So we have first your loss on drying. So loss on drying is the amount of volatile matter present on your compound. So, kasali na dyan si water, kasali na dyan ang mga solvents na naiwan. So, that's your loss on drying. Okay? And then, we have water content determination. So, sa water content determination, of course, specific na for the water content. No? So, we have 3 methods of water content. Ang official lang sa USP, take note, ang official lang sa USP ay method 1. No, method 1 is your Carl Fisher titration. Okay, so si method 1, pwede siyang 1A, 1B, 1C. So 1A is direct, 1B, no, 1B, so B for back titration or residual, no? Residual titration. And 1C, C for colorimetric. Ayan. So you have your Carl Fisher titration. The official water content determination method sa USP. Next, we have azeotropic distillation method 2. And lastly, we have gravimetric. No? So we have method 3. Okay. Sige, next, specific or special test. So we have the different specific impurities for some drugs. So example lang ito, for paracetamol, we test it for impurities on presence of p-chloroacetanilide. And for quinine, we have dihydroquinone sulfate. Okay, so tapos na tayo sa RMQC. Now, we move forward to in-process quality control. So first, for powders and granules. Okay. Okay, so for powders and granules, we check the particle size distribution. So the most reliable method is optical microscopy. Kaya lang very time-consuming ito kasi titingnan mo isa-isa, no, via the microscope. So the fastest would be the sieve analysis. No? So sa USP din, the method is sieve analysis. No? So sa USP, the acceptable method is this sieve analysis. So using your sieve. Okay. So we arrange your sieve, no, on varying numbers. So ina-arrange natin siya from largest pore size or largest smallest sieve number to largest sieve number. So the sieve number tells you the number of square openings per linear inch. So meaning sa isang inch, no, para hindi kayo malito, no, so sa isang inch, so if ever this is 1 inch, no, ilang box or ilang square openings ang kasya. No? So let's say sieve number 20. So meaning 20 boxes ang kasya sa isang inch. No? So as you would expect, the greater sieve number, the smaller the particle size. Kasi, di ba, for example, sieve number is 40, meaning 40 boxes ang kasya. So mas liliit yung butas. So, mas fine ang particles na dumadaan doon. Okay? So, that is your sieve number. No? The number of square openings per linear inch. No? So, per inch, ilang butas ang present. So, mas madami, mas fine. No? So, the greater the sieve number, the smaller the particle size na nasasala. So, kaya ina-arrange natin siya from smallest sieve number increasing. So 20, 40, 60, 80. No? So para yung mga malalaki, so tapos tala na po sa next na mas gamay ang magets naman. So yon. You have your sieve analysis. Okay? So sieving methods, we have mechanical agitation. Ipapatong mo lahat tapos i-shake-shake lang. So that's your mechanical agitation. Then we have air jet. So sa air jet class, isa muna sa 20 tapos may stream of air na mulusot sa sieve. Tapos pa. Okay? Tapos titingnan mo kung lulusot siya sa sieve. Murag nag-apply kag pressure ba? So for example, ito yung sieve. Maglalagay ka ng air papunta and so that's your jet. And then sonic shifting, same lang sa mechanical agitation pero hindi manual, no? May ganun-ganun talaga. May machine na gumaganon-ganon siya sa sieve. Okay, so that's your sonic shifting. Okay, so paano natin malilinis si sieve? No, 'di ba? Syempre butas-butas 'yon. So, 'di ba sa screen minsan merong mga nasasabit na powder. So, how do we clean? We can use air jet. So, same yung kanina, no? Magpalagay ka ng air papunta and pabalik para yung mga na-stuck na mga powder sa butas na matatanggal, no, by the pressure. Liquid stream, hugasan mo. So, high pressure na tubig para malinis. Diba? Mura na mo naglinis o saan. No? Diba? Minsan mahirap siyang linisin after using. And of course, gentle brushing. no? Gentle brushing. So parang ganun lang din yung sieve, literal, no? So ayan, pinaganda lang yung methods because of the manufacturing side. Ayan. So next, we have density. No? So we have different types of density. First, we have your bulk density. So bulk density, so that's the ratio of the mass of untapped. So isulat natin. So mass of untapped over the mass of tapped. Tama ba? And it's, no, total mass. Total mass. Okay. And its volume including the contribution of the interparticulate void volume. So we can use graduated cylinder, volumeter, and vessel. Sige. So sa graduated cylinder class, basically, you will measure, let's say, 10 grams. So sa graduated cylinder, constant mass. So you will use 10 grams of powder. Tapos, titingnan mo, volume dapat pala dito, volume. Erase. So, density is mass over volume. So, untapped ito, untapped. So, meaning wala kang tapping na ginawa. So, basically, yung 10 grams class, ibubuhos mo lang sa graduated cylinder. So, 10 grams, so automatic 10 grams na itong mass, yung volume, i-measure mo sa graduated cylinder. No? Ano ba yung volume ng 10 grams sa graduated cylinder? Yun lang yung bulk density. Sa volumeter, tsaka sa vessel class, this is constant volume. No? So, dito, we have constant volume. So, you will measure your powder kung how much ang kakasya sa volumeter. So, si volumeter class, na siya constant volume. Tapos, you will get the mass. No? So, dito, given na yung mass, 10 grams, titingnan mo yung volume to get the bulk density. Dito, may volume ka na, then you check the mass. Okay? So, yun lang yung difference. Okay, next, tap density. So, dito meron ng involved na tapping. So, kung graduated cylinder pa ni, itap ni mo siya. Itaktak ni mo siya. Kanina, lagay mo lang siya. Buo lang mo siya sa graduated cylinder. Yun na yung bulk density. Sa tap density, no, itatap mo siya for many times. Okay? So, that is the density attained after mechanically tapping a container with the powdered sample. So, we have 3 methods. So, wala nang volumeter. So, method 1 and 2, graduated cylinder. So, same pa rin class. Constant ang mass, no? Tapos titingnan mo lang yung volume change sa graduated cylinder, no? So syempre 'di ba pag mag-tap ka, mas compact ang powder kasi matatanggal yung air or yung void sa gitna. So yun yung tap density. No? And then we have vessel, constant volume. Okay? So, yan. Next, we have powder flow. So, diba, the powder flow is important for the tableting process of the powders. No? So, syempre, how it flows from the hopper, no, para magawang tablet. Okay, so first we have angle of repose. Ayan. So, angle of repose is the angle assumed by the cone-like pile of the material. So, we have fixed funnel, no? So fixed funnel, so it can have a fixed cone with constant diameter or a freestanding cone with constant height. Sige, so ang mangyayari class, no? We have your funnel. So, Ayan. So, maglalagay ka ngayon ng powder dito. And then, from that, your powder will form a parang mountain or cone-like pile of the material. So, now you will get the height of the cone and the radius, no, of the cone. Okay? So, you will now get that to compute for your angle of repose. So, angle of repose is equal to arc tangent of height over radius. No? Kung diameter ang given, divide nyo lang by 2. So, to get the radius. So, yung nasa ilalim, no? So, pag fixed cone, constant ang diameter. So, kahit na anong powder, ang diameter is the same. So, divide nyo lang by 2 si diameter. Pag freestanding cone, constant si height. So, ang pabago-bago lang is the diameter or the radius. No? So, using the two values, you can solve for the angle of repose. No? So, that's arc tangent or shift tangent. No? Sa calculator ninyo, shift tangent, tapos height over radius. Or pwede, isolve nyo muna si height over radius, tapos shift tangent. You still get the same answer. Okay. Next. So apparatus, you can use a fixed funnel. Yung sinabi ko kanina, we have tilting box. So sa tilting box class, same lang siya. Pero, lahi nagporma. No? So, we have a box. Pasensya na sa drawing. Tapos si box dire, naa siya'y buslot sa tunga. No? So, mo-tilt ang box, matapon ang powder, mag-form pa rin ng cone. Parang ganun lang. So, sa funnel, pag bubo ni mo, si powder mag-form ng cone. Sa tilting box, para siyang box na igaganon mo lang. So, pag yabunin mo sa box, mag-form gyapon o cone like powder. So, same lang gyapon. Measure the height and the radius. And then, we have revolving cylinder. So, same. Your cylinder will revolve. So, si cylinder mo revolve. Pag revolve niya, matapon ng powder, then mag-cone. So, same premise. Iba-ibang setup lang. Okay. So, angle of repose. So, anong maganda? So, maganda kay angle of repose, mas maliit na value, mas maganda ang flow. So, less than 25, excellent flow properties. 25 to 30, good. 30 to 40, fair. Malaki by 40, mas malaki sa 40, poor na. So, kung hindi nyo man ito ma-memorize, isipin nyo na lang, mas maliit, mas maganda. So, somehow, bana-bana na lang, no? Estimate na lang kung ano yung flow property. No? Yun. Sige. Next, we have Hausner's ratio. So, si Hausner's ratio class, it also tells you on the, it also pertains to the powder flow. So, dito naman, we use your top density and bulk density. So, sa Hausner's ratio, it's the ratio of the initial volume over the final volume. So, before tapping and after tapping na volume. Pero pag density ang ginamit nyo, baliktad. Top density over bulk density. Okay? So, sa Hausner ratio class, kung volume ang gagamitin nyo pag solve, yung before tapping na volume over after tapping. No? Initial over final. Pero kung density ang gagamitin nyo pang solve, top density, yung pinap na density over bulk. No, baliktad siya if density ang gagamitin. So that's your Hausner's ratio. So same class, the smaller the value, the better. No, the better flow property. So less than 1.25, good. 1.25 to 1.5, fair to passable. Greater than 1.25, poor. No, mas malaki, pangit. Next, we have Carr's compressibility index. So sa Carr's compressibility index class, somehow same lang siya, but in percent. No? So sa kanina, initial minus final over initial times 100. No? So same, murag pa-expand lang siya na Hausner's ratio. So top minus bulk over top times 100. Okay? So same premise, greater value, poor flow property. No, mas maganda, mas maliit. 5 to 13, excellent. Okay? So, ayan. So, sa angle of repose, Hausner's ratio, Carr's index, the smaller the value, the better. Okay? Next, we have uniformity of moisture. So, we have coning and quartering. No, that's the method of checking for the uniformity of the moisture. So, for moisture content, dapat hindi lumampas ng 31 to 35 at first. No? So, the primary moisture content of powders and granules is 31 to 35. So, after granulation, no, the final moisture content dapat mas maliit na lang siya. So, ito, una, initial, 31 to 35. Pero sa final, dapat sobrang konti na lang. No? 0.55 to 1%. Okay? So, ayan. So, from powder, magiging granule via granulation. No? So, wet granulation is the most ideal. But then again, the moisture should be controlled. Okay? So, this is summary ng ating powders and granules in process quality control. Yan, Hausner's ratio, porosity. So, hindi natin na-discuss si porosity, no? So, basically, bulk volume minus the top volume times 100 over the bulk volume. Then, sa moisture content, yung Carl Fisher and your content uniformity. Okay, next we have your tablets. So for tablets, in-process quality control is your hardness tester. So our hardness testing. So that's your crushing strength, no? So the ability of your tablet to resist abrasion, breakage, under storage, transportation, handling before usage, no? Na dili siya ma-crush, dili siya mabakli, no, dili siya masira on transportation and handling bago pa siya mapunta kay patient. So, the hardness affects disintegration, dissolution, and bioavailability. Ayaw din natin na sobrang hard ang tablet kasi minsan mahirapan na siyang mag-disintegrate. So, matagal ang disintegration, matagal ang dissolution, matagal siyang ma-absorb ng tao. Now, what if for pain siya? No? Pain reliever. Diba gusto natin na ma-relieve agad yung pain? So, dapat the hardness should be controlled. No, before wala pang machine for hardness testing, we use the rule of the thumb. So we use your itong finger, middle finger, and your thumb. No, so yan, i-crush ra ninyo ang tablet. Kung kaya ninyo i-crush, then it's not that hard. No, so pero very subjective siya. Kasi what if strong lang jud ang tao na nag-crush? No? So, ngayon, meron na tayong instruments for hardness testing. No? Before, rule of thumb. Yun. So, i-crush mo lang siya with that. Okay. So, these are our instruments. So, keywords lang tayo, class. Keywords. Pag may spring, Stokes Monsanto. Pag may air pump, Strong Cub. Pag pliers, no, that's your Pfizer, no? P, pliers, P, Pfizer. Then Erweka, your suspended motor driven with, medyo mahaba. And then kay Schluniger, anvil, no? Anvil. And si Schluniger, siya din yung most widely used, no? Si Schluniger, siya din yung most widely used. Okay. So, criteria, ano ang ating specifications for hardness testing? So, for plain uncoated tablet, we have 4 kilograms. Okay. So mga normal tablets, 4 kilograms. Si buccal tablets, no, 'di ba? Para saan si buccal tablets? For slow release. So gusto natin slow ang release, slow ang disintegration. So of course, gusto natin mas hard. No, 'di ba? Pag hard, again, matagal mag-release, matagal ma-dissolve. So gusto natin matagal ang effect. So they are generally higher in hardness. Sa chewable, sublingual, gusto natin mabilis, no? 'Di ba? Sublingual fast release of the drug. So 2 to 3 kilograms. Sa chewable, of course, 'di ba, di ta gusto mabungi ang atong pasyente sa pag-chew sa tablet. So dapat dili siya kaayo hard, no? 2 to 3 kilograms lang. Kaya ra sa ngipon i-chew, no? So you have your hardness test. Okay? So dapat hindi lumampas dito. Okay. Next, tablet thickness, no? Tablet thickness. So bakit natin kailangan i-control si tablet thickness, no? For aesthetic purposes. No, 'di ba? Pangit kay tan-awon ang tablet na baga. So we control the tablet thickness. So usually this depends on each company. No, kung unsa ka thick ilang gusto na ilahang tablet. Oo, para dili pod sakit itulon. Oh, pwede pod. O dili siya lisod ipack. No? For example, 'di ba sa mga foil, lisod siya if very thick ang tablet. Okay, so here we use your vernier caliper, no, to measure the thickness. So for example, si Unilab, sabi niya dapat ano lang ma'am, at 2 inches thick, or baga na ba, 1 inch thick, 1 inch. So dapat plus minus 5% sa 1 inch. No, yun lang ang ating range na pwede na thickness. Sa man of class, pag mag-duty na mo, usually sa mga manufacturing company, they have what we call 5-in-1 instrument. No, 5-in-1. Bakit 5-in-1 na siya? So sa isang instrument class, ma-measure na niya yung hardness, yung thickness, yung weight, tapos iba pang test ng tablets, no? So somehow ano siya, time saving, and di pod kalas og tablet na i-test. Kaya syempre isa lang ka tablet or pila lang ka sample, isahang testing na tanan. Dili siya paulit-ulit. So hopefully in your manufacturing duty makatry mo. No? So usually magod pag QC testing kamo ginapabuhat sa testing, no? Kung pasa or bagsak ang tablet. So next, sinabi ko na, no, tablet hardness, tablet thickness, tsaka tablet weight gina-measure pod. So the tablet weight, no, so that's the unofficial weight variation test according to USP. So basically you measure 20 tablets class, you weigh 20 tablets, tapos get their average weight. Now, if the average weight is less than 130, then the maximum difference is 10. Pag the average weight is 130 to 3 to 4, then 7.5. Then, if greater than 3 to 4, for example, 500 milligrams, then the maximum percent difference is 5%. So, let's say ang average weight niya ay 500 milligrams. 500.50 milligrams. So, multiply nyo ito by 5%. Tapos, i-plus and minus nyo kay 550. So, let's say, wait lang, kuha ako ng calculator. So, 5% lang, no, kay greater than 3 to 4. So, 500.50 times 5%. So, that is 25.025. So, idagdag nyo lang siya and ibawas sa 550. So, the weight of your tablet should be not less than, so, minus 550. So, 475.475 until 500 0.25. So, dapat hindi lumampas sa range na yan. So, pag lumampas ng 2, bagsak. No? I know. No more than 2 tablets are outside the percent weight variation. So, dapat hindi lalampas ng 2 ang not more than 2 ang lumampas sa weight na ito. Okay? Kung lumampas ng 2, so say for example, naamoy tablet na 550 milligram, 550 milligram, and meron kang 400 milligram, tapos naapa dyan isa na 390 milligram, then bagsak na dyan siya. No more than 2 tablets are outside this range. No? So, depende sa weight ng tablet, kung ano yung maximum percent difference, tapos i-plus minus lang ninyo. So, multiply the 5% sa total or average weight, then i-plus and minus. So, i-minus para makuha nimo ang minimum limit, and then i-plus para makuha nimo si maximum limit. Okay. Next. So, we have content uniformity. Weight variation class if the active ingredient is 50 milligrams or more. No? Pero pag less than 50 milligrams, we do content uniformity. So sa content uniformity class, i-check yun mo ang sulod per tablet. No? So sa weight variation manggod, ang premise is kung pare-pareho ng weight, pare-pareho silang sulod. No? Pero kung gamay ra kayong active ingredient less than 50, dili somehow proportional ang weight sa active ingredient. Nagets din. No? So sa weight variation, ginagamit siya if 50 milligrams or more ang active ingredient. Meaning sa isa ka tablet, ang active ingredient 50 milligrams or more. So, you use weight variation. Kaya ang claim sa weight variation, kung pare-pareho ng weight, hindi magkakaiba ng weight, pare-pareho sila ng laman, no, na amount. However, kung very minimal yung active ingredient, let's say digoxin. Si digoxin is naka-nanogram. No? So, kasi konting-konti lang si active ingredient doon, hindi ka sure. No? Hindi ka sure na kung pare-pareho sila ng weight, pare-pareho silang sulod na digoxin. So, in content uniformity, you really check each tablet. No? So, sa 10 tablets, you check kung 10 nanograms yun ba siya or 500 nanograms yun ba siya. No? So, it should be 85 to 115% of the labeled claim. So, let's say ang labeled claim is 500 nanograms. So, dapat hindi siya lalampas sa 85 to 115% of the 500 nanograms. If bagsak, then go to stage 2. Plus 20. No? Tapos ito na ang acceptance criteria. Okay? So, ayan. So, i-test mo talaga isa-isa. So, weight variation, i-weigh mo lang. Then, check mo yung weight. Kung may mga malayo yung weight. Dito, i-check mo talaga yung exact content. No? Kaya siya content uniformity. Yan. And then, we have your finished product quality control. So, first, 4 tablets. So, for tablets, identification, assay, and next, friability. So, yung identification class, yun yung mga sinabi ko kanina. Pwedeng IR, chemical, physical. For assay, pwedeng different assay. No? Instrumental, biological, etc. So, next specific test is friability. So, sa friability, no, that's the percentage lost by the tablet in packaging and transport. So, sa hardness kanina, no, ability to resist breakages, no, mabuak, masira. Yan. Pero sa friability, that's the percentage lost, no, by the tablet na weight. So, yun. Di ba si tablet, pag madugay siya, sige siya ka-shake, pwede na mag-shed off ang powder sa iyaha. No? So, makulangan siya weight. So, that's your friability. No? So, the instrument used is your friabilator set at 25 revolutions per minute times 4 minutes. So, 4 times 25, kaya 100 rotations siya. So, 100 revolutions, all in all. Okay? So, you check 20 tablets or kung greater than 650 milligrams ang weight ng tablet, weigh ka ng tablets na equal sa 6 to 6.5 samples. Okay. So, unang-una, i-weigh mo sila, yung tablets. After nun, ilo-load mo siya sa instrument. So, si instrument class, wala ko yung picture, bale, magbalik-balik siya. No? It's a tumbling apparatus. Mura siya Ferris wheel, no, na paspas. So 100 ka tuyok, no? So after magtuyok siya 100 times, you weigh it again. So that's the final weight. So initial, before nyo nilagay sa friabilator, minus the final over initial weight. So times 100. So acceptance criteria is not more than 1%. Dapat less than 1% ang lost na weight. So 0.9, 0.8, no? Pero if it's a new formulation, ang ating limit ay 0.8. So dapat hindi bumaba. Hindi lumampas sa 0.8 ang percentage lost, no, na percent friable. Okay, what if bumaba? So you can do stage 2. So sa stage 2 class, dagdag ka ng another batch. So dagdag ka ng 20 or 40. Dagdag ka ng 2 trials. 2 trials. So i-average mo yung 3 trial, dapat hindi siya lumampas ng 1%. So kung lumampas pa rin, dun pa bagsak. Pero kung hindi lumampas, then pasa siya on the second trial or on the second stage. Okay, take note, it should be without capping and shipping. No, without capping and shipping. So, what if ma'am, pumasa siya dito pero nag-capping and shipping? Bagsak pa rin. No? Bagsak pa rin. Okay? Sige, so stop muna tayo dito na part. Wait lang ha. Kasi I think na-time yung piece nyo at 9:15.
Recording stopped.
Sige, so at 9:15, ma-upload ang quiz. Sige.